Welcome to Ask the Expert. It's a brief, informative, and lively discussion with experts in the type 1 diabetes field and related interdisciplinary research. We're recording this event and we'll post it on the Sugar Science site YouTube channel shortly after the presentation. If you have any questions for our guest, Nayara Leite, today, please feel free to enter them in the chat or raise your hand at the end of the presentation. And so it's my honor to introduce Nayara Leite, PhD. She's a postdoc based currently in the Harvard lab of Doug Melton. She's originally from Brazil, and I'm going to let her share what she's doing currently. Welcome, Nayara. How are you?
Thank you. Thank you. First, thanks for the invitation. It's my pleasure to be here. Today, I will talk a little bit about the paper that we published last year and how I'm thinking about the next steps of my research. So I have a few slides. I will start sharing my screen. I think you need to enable that. Yes, perfect. Yeah. I think now you can see my slides and I have the chat open too. So if you have any questions during my presentation, you can just type in the chat or you can unmute yourself if you want and just speak. All right, so today I'll talk about how we are using induced pluripotent stem cells for type 1 diabetes modeling in vitro and how I'm thinking about protecting these beta cells. I think as everybody here is familiar with, type 1 diabetes is an autoimmune disease in which the beta cells are distracted by the immune system. And we know that genetic and environmental factors may render the beta cell the susceptibility to be destroyed by the immune system. However, we don't know exactly what triggers the disease and the progression. So I think there are two major components in the disease initiation. One is the beta cell vulnerability, and two is the immune system dysregulation. And what I mean by beta cell vulnerability is just the risk of the beta cell to be attacked and destroyed by the immune cells. Since in the Melton lab, we can generate a lot of cells, billions of cells, my research is focused on the beta cells. In this picture, what I'm showing you is a representation of a beta cell. And as you know, the beta cells are the cells that produce and secrete the hormone insulin. And insulin accounts for up 50% of the protein production on the beta cells. And this can increase in response to metabolic demand. For this reason, the beta cells are prone to ER stress. ER stress develops when the demand to synthesize insulin exceeds its capacity. And this situation can arise as a variety of different perturbations. And specifically in type 1 diabetes, can be caused by high metabolic demand or high glucose levels. The resulting perturbation in the beta cell homeostasis can lead to unfolded protein response that increase these misfolded proteins and can then lead to apoptosis. And I don't know you're familiar with, but beta cell stress has implications for immunogenicity in diabetes, in type 1 diabetes. And then this impaired protein process, insulin protein process, can lead to generation of antigens. And these antigens can be presented by the HLA molecules. Not just that, beta cell stress can also lead to local release of chemokines that can attract the immune cells. And not also on top of all these things that I just described, viral infection, reactive oxygen species, and also inflammation are related or can be related with type 1 diabetes onset. And what all these things have in common is that they have beta cell stress as an initiation factor, suggesting that the beta cell stress may be a common factor for the disease. So one thing that I'm particularly interested is studying beta cell stress, and we will talk a little bit more about it later. So in the Melton lab, the main goal is using beta cells for cell replacement therapy. And what we do in the lab is generating beta cells from either iPSCs that are induced pluripotent stem cells or embryonic stem cells, the ESCs, with the goal of producing glucose responsive insulin secreting beta cells that will go at some point back to the patient. And what it's really important is that this production needs to be scaled in a high number. As you can imagine, there are so many people with diabetes, so we are able now to have a process in which we can produce a lot of cells in larger scale. And in my view, there are two main challenges with beta cell replacement therapy. The first one is necessary to have functional integration of those beta cells to provide glucose homeostasis and durability of these engrafted beta cells over time in the host or patient. And second, is necessary to have immune evasion. So since type 1 diabetes is an autoimmune disease, as you can imagine, after you put back the cells in the patients, it's likely that the immune system will recognize, like in the first place, the beta cells that we just transplanted. So in order to study these interactions that may occur in type 1 diabetes, in human type 1 diabetes, my colleagues and I developed a model to study the immune interactions that may occur in type 1 diabetes, human type 1 diabetes. And as you can imagine, type 1 diabetes is really complicated. And until now, we don't know a lot of things about it. So it's hard to find a good human model that would recapitulate all the complex features in the immune interactions between the immune system and the beta cells. So then we decided to focus on the interaction of the beta cells with the T cells, which we believe that is one of the main factors on type 1 diabetes triggering. So in this figure, I'm showing you the experimental approach that we use. So basically, we generated beta cells from induced pluripotent stem cells from patients. And since we are working with living patients, we were able to isolate the peripheral blood from the same patients. And then we isolated the PBMCs, or the peripheral blood mononuclear cells. And then we performed co-culture assays with the goal of understanding and studying what are interactions that occur between the immune cells and the beta cells. And one thing that is important to me to mention here, in order to recapitulate the type 1 diabetes autoimmunity, we found that the beta cells had to be stressed prior to the co-culture, which is in line with previous studies that have shown that beta cell stress is linked with autoimmunity in type 1 diabetes. So now I will talk briefly about what we learned from this study. So the first thing that we learned is that the T cells are activated when they are co-cultured with autologous stressed beta cells. And the way that we measured this was using different readouts. And first, I'm showing you here cytokine secretion. And if you look here on in both graphs, I'm showing interferon gamma and IL-2 after co-culture with type 1 diabetic beta cells stressed induced by tapsigardin, we have an increase of interferon gamma and IL-2. Another way to look at the same result or another readout would be looking at activation markers. And we also observed that after co-culture, we have an increase of activation markers on the T cells that were co-cultured with stressed iPS-derived beta cells. So then our question was if this T cell activation is mediated by direct T cell HLA interaction, or in other words, if we avoid the T cell to bind to the beta cell, would the same effect happen. And in order to test that, we use 3 different approaches. The first one is a classical immunology approach, where we use anti-HLA blocking antibody. So then the T cell could not touch the beta cell. The second way that we did this was using a transwell system, in which the T cells are in suspension, so they cannot again touch the iPS beta cells. And the third way was a genetic approach, where we downregulate the HLA class 1 on the beta cells. So then the TCR would not recognize anything on the beta cells. And what we found is that using all the approaches, when you prevent direct T cell HLA interaction, you have reduced T cell activation that is represented here by the white line, and here and here. So what we learned from this experiment is that this T cell activation induced by the iPS beta cells is mediated by TCR engagement. So then, as you may know, the beta cells are not the only cells in the pancreatic eyelids, but they are the only ones that are destroyed. So we were wondering if this could be also recapitulated in vitro. In order to do that, we not just generated iPS-derived beta cells, but we also generated from the same donor iPS-derived alpha cells and iPS-derived cardiomyocytes. And then we were wondering if we would see a higher activation against the beta cells, just like what we see in the disease. And in order to do that, we performed the same experiment that I described to you already, co-culturing the cells and then looking at activation markers and cytokines release. So here, what I'm showing you is just one activation marker. And on the top one, you can see the percentage of cells that get activated against the beta cells. Here is the percentage of the cells, the T cells that get activated against the alpha cells. And then in the bottom one, against the cardiomyocytes. So we can observe there is a much lower activation against other cell types in comparison with the iPS-derived beta cells. So in summary, this in vitro platform that I just described can be used to understand the cellular and molecular components relevant for immune interaction in human type 1 diabetes. And in addition, we can think about using the system for testing for immune protection of tolerogenic iPS-derived beta cells before cell replacement therapy. And finally, this model can pave the way for other disease models that take advantage of patient-derived cells and to study the outcome of autoimmune disease. So now that we have the system and we have the platform or the cells, the next step for our lab is immune protect the beta cells. And as you can think about it, there are two main approaches. The first one would be using an encapsulation approach, that it's a physical barrier that would just block the immune cells to touch the beta cells. And the second approach would be a naked cell approach. And the naked cell approach is the one that we are more interested in. So here is a summary of some ideas that people in the lab are pursuing. So basically taking the lessons that we learned from cancer immunotherapy and from pregnancy, we are kind of trying to generate these modifications on the beta cells and see if we can protect them from immune recognition. What I am most interested in learning is actually from the beta cells lessons. And you understand what I mean by that by just a moment. This is how I see the type 1 diabetes pancreas. I think it's like a war between the beta cells and the immune system. And the researchers are trying to figure out what triggers type 1 diabetes for so many years. Or in other words, who gave the first shot when everything that you can see is just a war and destruction. Maybe we can look to this problem in a different way. So instead of studying what trigger, I want to study what can protect the beta cells from dying. Thinking about beta cell replacement therapy. So here is a study from the medalists. And medalists are patients that have diabetes for over 50 years and they still have functional beta cells. And what they observe in this study is that there are different beta cells that are left. So what I mean by that is there are some beta cells that they can be found outside the eyelid. There are some pancreatic eyelids with few beta cells. And there are some eyelids that have many beta cells. So my question is, what makes this beta cell here in the category C be alive compared to these other ones that are not here anymore? So what I'm trying to understand is what is the difference of the beta cells that are stronger and still alive versus the beta cells that die. The best experiment would be just trying to figure out from these patients what is the difference. But as you can imagine, it's really hard to get the samples and the patients is still alive. So in order to do that, I use alternative approach. That is, I'm working on sequencing pancreatic islets from recent diagnosed type 1 diabetic and non-diabetic patients. So then my idea is understand what is the differential gene expression in beta cells from recent diagnosed type 1 versus non-diabetic beta cells during the trigger. And using data published from the Human Pancreas Analysis Program, I was able to analyze what it was already out there in comparing the non-diabetic versus the diabetic beta cells that from recent onset diabetes. Not surprisingly, I saw that a lot of genes that are upregulated on the type 1 beta cells are related with immune recognition and beta cell stress. So again, in line with what we observed with the first part of my talk. So what I'm doing now is working on modulating these genes of interest on the S beta cells or the induced pluripotent stem cells, derived beta cells. And as I said, these genes are related with these markers, let's say peptide presentation, cytokine signaling, and beta cell stress. So my idea is using a target library in which I will downregulate the genes that are being upregulated on the stressed beta cells and see if we can conquer protection, 1, against stress, and 2, against immune recognition. So now the questions that we still have is how many genes need to be silenced to have protection? And how many genes can we change from the beta cells in order to keep the beta cell being a beta cell? Or in other words, keep being functional. So, yeah, as I said, the next steps is protecting or finding a way to protect the beta cells from autoimmunity and also alloimmunity. And then finally, the long-term goal would be then transplant the cells into patients. And here is just a picture of the Melton Lab before COVID, where everybody was without masks. And I want to thank you all, my collaborators, the Melton Lab, Doug, for being an incredible mentor, and all the founding sources. And I'm happy to take any questions.
That was great, Nayara. Thank you very much. You really brought us through a lot of information and data in a very quick time frame. So thank you. Is anybody like to submit a question into the chat or ask a question? Unmute yourself. Okay, I will ask.
Yes, yes,
Oh, yeah.
I have a question.
Yes.
Thank you. Great presentation. Very interesting. I was a little bit curious about the beta cell stress caused by viral infections and those kinds of things. And I was just wondering, for people that are at risk to get type 1, wouldn't it be smart to, like, for example, when they get sick, when they get COVID or something like that, can you try to lower the beta cell stress for those people, you know, a shorter amount of time, just to reduce the risk of getting the onset of type 1?
I think that's a great question. I don't know for sure if you could do that, but probably if you could pick up the time that is happening, and if you could give some kind of immunosuppressor, I think it would be possible to avoid, or at least to kind of interrupt a little bit the progression. I don't know how effective it is. I know there are some clinical trials with like blocking antibodies against the I think CD3 or like some specific T cells, but in the end of the day, people still get diabetes. It's just that it takes longer.
Okay.
So if we could figure out that, I think would be a great thing. So not just messing up with the immune system, but if we could figure out something that would just keep the beta cells alive longer, would be really nice.
And high blood glucose is causing beta cell stress.
Yes, correct. Okay. Yeah.
Thank you.
There's a question from Alex here. What made you decide to use the naked cell approach over the encapsulation approach? Does it have any distinct advantages when it comes to protecting beta cells?
Yeah, that's a good question. So I think mainly because any organ, the pancreatic islets are highly vascularized. So if you think about it, if you have a naked cell or without any, let's say, material around that, the chances of this new cell product or transplantation graft to be vascularized and receive a better nutrients, oxygen, to avoid hypoxia. I think that's the main advantage, to provide better engraftment. Another reason why we chose the naked cell approach is that you can play with many things. So I talk about a little bit about the things that we are doing in the lab, thinking about HLA or stress in my case. But if you think about the naked cell approach, you can do pretty much everything. So it's like, I think there is a lot of things that we can be done in the naked cell approach yet, that we are just starting. And of course, this would be a next generation product that maybe will be, let's say, released like many years from now. Probably the first product would be with a encapsulation device, just for safety reasons.
He said, thank you very much. I had a question for you. You know, when cells get into ER stress, the PERK, the protein kinase RNA like ER kinase, can be involved, and there's an abnormal reduction in protein synthesis rates. Do you guys see that sort of stutter, like when the beta cells start having their stress? Does the protein synthesis rate start declining as a result?
That's a good question. So I think the short answer is yes. I've never followed up over time. So I think an interesting experiment would be just starting inducing stress and then collecting the cells in different time points and see how the function declines, as you were saying. What I did was, let's say, before stress and after stress. And I didn't show you here. I tested different stressors like cytokines, ER stressors, high glucose, a combination of those. And I see that the cells are functional before stress, but then after you induce stress, the functionality is gone.
Hmm.
But all the stressors that I used, they were pretty harsh. So I think would be interesting to see how it would develop over time.
Nayara, what's your thoughts on this new work that's coming out of Anil Bhushan's lab, that the beta cells are actually becoming senescent, and it's the NK cell deficit that's not allowing for them to be cleared from the eyelid, and then that's engendering the immune response or adding to it. What do you think? I mean, that's really new, and I think there's a really divided, perspective on it.
I don't know. I think it's really interesting. I can see. I've read a little bit about senescence before, more related with type 2 diabetes. I don't know exactly what are my thoughts on the type 1 field. I don't know how fast the cells would get senescent, but if this is, I think probably it's also a contribution. And maybe there is like different types of type 1 diabetes. I always think about it. I don't think there is one type of type 1 diabetes. So maybe some type 1 types would be more related with senescence and decrease of NK cells. Some others would be related with other things. So I think if we could figure out a way, if senolytic drugs would like avoid type 1 diabetes, or like if we could combine like different approach, I think it would be nice. And just to wrap up this question, I don't think there is one solution for the problem. I think it will be a combination of different treatments that will finally get to the cure, in my opinion.
That's a very educated opinion, and I think everything you said, this is totally right on, because it's like it is totally a heterogeneous disease in its like pathway to get there and its presentation. Even when you showed the medalists, they had a heterogeneous presentation. So it's like such a smart way of thinking about it. But someone, Anna Margarita, has asked, thinking about transplantation into patients, would you transplant only the naked SC beta cells or also another pancreatic cell types like an alpha cell?
Yeah, that's a good question. And this is one question that we are working in the lab right now. So I don't have yet a clear answer for you, but I can tell you what we are doing. So basically, since we can generate not just beta cells, also alpha cells and like delta cells, we are building eyelids where we can control the ratio of cell types that are in each eyelid. So for example, we have 50% alpha and 15% beta cell in one cluster, and then another cluster that is just beta cells, and so on and so forth. We know how important is the paracrine communication between the alpha and beta cells to promote glucose homeostasis in vivo, but we still don't know if this would be the best approach when you are thinking about treating the patients. Since the alpha cells are left on type 1 diabetic patients, maybe just the beta cells would solve the problem, but we still working on that. But that's a great question, and we are thinking about this for a long time.
Yeah, you guys are one of the front runners in this race to get an eyelid implanted. And yeah, it's a real difficult question. I've been watching and talking to Ekaterine Bershvili at University of Geneva. She has some really interesting approaches using human amniotic epithelial cells and creating the organoid, right, to do the implantation there. So who knows, maybe some sort of collaboration between these different camps will drive it to the finish line. But I wanted to, there's another question. Could you co-culture with senescent cells as well and see how they behave both with non-diabetic and diabetic people? Interesting.
You mean co-culture the immune cells with senescent beta cells?
Sorry, so I was just wondering, this new theory with the senescent cells being, like blocking, for the new cells to be produced. I was thinking, can you take the immune system from a diabetic versus a non-diabetic person, try that against senescent cells and see if the non-diabetic person, the immune system clears out those senescent cells or not? That makes sense.
I think probably you could try to reproduce something using the model that we use, using stem cell derived beta cells. I don't know, how would you induce senescence? probably there is a way. In vivo, I know that there is some really good models for senescence in vivo, but I don't know exactly in vitro. But I think that's an interesting way to think about it. Yeah.
Yeah.
Thank you.
That's a really interesting idea. Maybe time for a second postdoc, Henrik.
I know.
Or a third, or where I'm not sure where you are in your career. You look young. So I also wonder, you know, maybe this might be the last one because I know you're super busy, Nayara. This has been really fascinating. But are you guys following the methylation, you know, markers or the methylation profile of your cells as you know, go from iPSCs into the alpha cell, the beta cell, or whatever? Are you careful with that, or is that like something you don't worry about?
So Juan from our lab, he published a paper, I think in 2019 or 2020, now I don't remember, where he followed the methylation profile from the pluripotent state until the beta cells. He used embryonic stem cells as a model, and what he saw actually was that what was expected to have open chromatin was open for gene transcription, and what was supposed to be closed was closed. But what was really nice about his findings is that he was able to find genes that are known as clock genes that can regulate metabolism on the beta cells. And then the final part of his paper, he from this methylation data, he was able to manipulate the clock genes on the process of differentiation, more specifically on the last stage where they are already beta cells. And then he was able to make better beta cells. So I'm not too familiar with what is the epigenetic signature of the cells that we make in the lab, but I think that's a really important question. And if any of you guys are interested, we have the resource posted online. So you can go online and check what are the profile of like the mutilation or any other candidate that you like during the stage of differentiation.
Yeah, no, I knew about that paper. That's a very cool work. I just wondered, like, when you're taking you know, when you're bringing a person's cell and you know, going them back to the stem cell and then bringing them forward into a beta cell, like, do they have any, you know, bookmarks basically, right, with the methylation? So it's just, that's the kind of was thinking about, and I hadn't really seen anything about that. So that's what I was wondering.
Yeah, I think probably yes. We know, like, if you look at the methylation profile, comparing iPSCs versus ESCs, they are different.
Yeah.
There are like a lot of differences. So probably if you go up to beta cells, you will also see differences. And maybe they are important. We just don't know. Yeah.
Another postdoc project.
I know. You are giving so many ideas for projects. I wish more people from my lab was here so that they could do it.
I know. Oh boy, yeah. There's just not enough time to do everything. You have to do what you can do. But yeah. Does anyone else have any other questions? This was fascinating. And I applaud you, Nayara. You're really doing some beautiful work. It's so detailed, and it's also very clear, very, you know, your hypotheses are very well supported. So it's great. Thank you again for talking with us.
Thank you.
Wish you the very best, and we'll be looking for your next paper.
Thank you. And if you have any other questions, please feel free to send me an email, and I'm happy to chat more.
Absolutely. Yeah. Or, you know, reach out to her. I think you're a collab member on the sugar science, so maybe get you there too. Thank you again. Have a great rest of your afternoon.
Thank you. You too.
Bye-bye.
Bye.
Bye all.